Pleasure to introduce Richard Heller. Richard is a professor at the Old Dominion University in Norfolk. Richard is one, I would say, even the key founder of the technology of using electric field for DNA delivery into tissues. First it was small molecules, then it was tissues. And he has always been very active and inventive in that field, so I'm very happy to have him here to present the new result on in vivo electroporation to the heart.
Okay, thank you, Danielle, for the introduction, and I thank the committee for inviting me to give the talk. So, as Danielle just said, I'm going to talk to you about our work in using gene electrode transfer to deliver plasmid DNA directly to the beating heart. And let's see how it works. So most of you know, just a very brief introduction, coronary heart disease is a major issue, particularly in this country with a large number of deaths. And some of the issues are where vessels are blocked, as you can see depicted in these two pictures, and what happens is obviously restricted blood flow, and you get ischemic areas within the heart, you can get damage to the tissue, and then an infarct forms, and then usually if it's not taken care of in some way, that you're going to end up with the heart stopping and death. So the idea that we were looking at, and many others have used this with different approaches, is to try and see if we can induce some either new vessel or elongation of existing vessels into that area to be able to reperfuse. Oh, I'm sorry. You know, this clock didn't start?
No, but we know.
Oh, you know when it did. Okay, I thought maybe I can go as long as I want, but that's okay. So in any event, so if we could do that, the thought would be is maybe we could either reduce the infarct and the impact of that lack of perfusion into that area, or actually eliminate it completely. And so our thought was to be able to do this in a way to deliver angiogenic factors into or around that impacted area to be able to get flow in there. And one of the things that we looked at using electrode transfer is because, at least in our opinion, is that you can have better control over the delivery and the expression profile by doing it that way. So you can, by manipulating the parameters, you can look at different amplitude of the expression as well as the kinetics of expression. And when dealing with angiogenesis, it's important to modulate that so that there's the right amount and not the vessels don't overgrow. So the first model that I'm going to talk to you about is a swine model, the porcine model. And in this case, what we do basically is open up the chest, expose the heart, and then what we do is tie off the LAD, and we form an ischemic area. And this is a permanent tie-off, so we don't just tie off for a small amount of time and then release the flow, but we keep it that way. So we never release that tie. The other aspects of this that we're looked at, and if you ask me why we picked 4 sites, I don't have an answer for that. It just happened that way. But in any event, what we do is we pick 2 sites along the border of between the ischemic area and the non-ischemic area, as well as 2 in the ischemic area, to try and get that reperfusion in that area. And we tested a variety of different electrodes to try this, and what we ended up settling on is this 4-needle electrode, and this 5th needle right there is actually an injection needle. So one of the issues that we found early on was that because, unlike a lot of other tissues, when you inject, making an injection in a site, you could see blanching or some swelling of the tissue, and you know exactly where you inject it. In the heart, this wasn't the case. So if by putting the injection port in the middle, we knew exactly where our DNA was, and it was within the field. If it was outside the field, we wouldn't get much delivery, if any. The other things that I will tell you as we did this, we basically look at the perfusion of the heart prior to the tie-off, and we do this with an instrument called the SPY, and we inject fluorescent microspheres in, and it takes a 30-second video of it, and you could quantitate how much perfusion is in the heart. Then we tie it off. We do another video to see the difference between prior to post, and then we do our treatment, and then we wait a couple of weeks, at least initially, the initial experiments, wait a couple of weeks and then see what happens. So this is an example of one of the experiments. This is a swine. The chest is open. This is the surgeon actually injecting the DNA into one of the sites. And then this is after the injection and the electrotransfer procedure. This is the area that was treated. And you can see there's really no sign of any kind of damage to the heart, at least visibly. So we were pretty pleased with that. The one issue that we did have, of course, though, is that applying electric fields to the heart doesn't necessarily make the heart feel happy. And so on the first pulse, if you don't do it properly, the heart will fibrillate. And so what we had to do was to synchronize the pulses with the rhythm. And this can be a challenge depending on what the heart rate is, but essentially what you need to do, this is your refractory period where you need to be in. You want to start as close to the leading edge of the R wave as possible, and your pulsing needs to be absolutely done before the initiation of the T wave. If any of the pulse is still there at the start of the T wave, the pig will fibrillate. So this is, again, just a depiction of that injection and the pulse, and actually this has worked pretty well. Without this, they fibrillate every time. With this, it was a very, very rare occasion if they did. So looking at expression with this is looking at delivery of VEGF-A. And what was interesting with this, and this is in a healthy heart, we tested a variety of different conditions. We chose 20 milliseconds because of this restriction of having to be within that QRS period. We can't go much longer than that. We originally thought about doing longer pulses and lower voltage, but it turned out our voltage didn't have to be very high anyway. We were under 100 volts in this case. And each of these were pretty consistent in the expression. All were above injection only or no treatment. So we were pretty pleased with that. So the next thing to do was, as I was mentioning before, is to do a tie-off and to get an ischemic area, and then do the same thing again with the 4 treatments and look again at whether or not we can get expression within the ischemic area. As you can imagine, within the ischemic area, you're going to have dying cells, and the question would be, can we get expression within that area? So this is what it looks like. So this is a healthy heart. The more yellow or red, the more blood flow. And then this is right after tie-off. And you can see this whole area now has no perfusion in it at all. And these ties represent where we would treat. And then this is again delivery of VEGF-A, and then looking at over a 2-week period. And then again looking at these different pulse conditions, and then looking at expression within the ischemic area and within the healthy area. And it actually is fairly interesting. Well, I can't read that. But, so if we look at 20 milliseconds, 60 volts, which is here, it's actually pretty interesting when we see this, that there we get a pretty consistent expression over the, at least for the first week, and then it drops off by 2 weeks, which is kind of the pattern that we were looking for. We only wanted expression for a short period of time. And consistently you could see that the expression is somewhat better in the ischemic area than the non-ischemic area, which is also was very interesting in there as well. So with this, we decided, okay, let's see, it's nice that we can get expression, but what can this actually do? And I don't know if this is going to work. These are, maybe it's not. I'm pointing at it like it's going to do something, but maybe not. If this did work, what it would show you is that in this one, will show you the perfusion. You could see the microspheres coming in. It's actually kind of nice, but no, maybe not. If you guys clicked on that, would it work? Okay, no, yeah, I know, but these are videos and they're not working. But, okay, so we'll just move on. Okay, so this is what you would have seen in the video. So again, you see in a healthy heart, you have perfusion through the whole thing. We tie off, then we treat, and then this is 2 weeks post-treatment. And you could still see the sutures here, and as you could see that perfusion is coming back into this area. And this is only 14 days after treatment. So we're pretty pleased with seeing this. One, all the pigs basically survive this tie-off of the LAD and this massive area of ischemia. None of them basically had a heart attack. And second, we're able to see in the treated pigs reperfusion into this treated area. And the nice thing about this SPY equipment is that there's software that you can use which would compare these three and actually give you numbers as far as the percentage of this area that's perfused, percentage of this area that's perfused, and then the percentage of this area. So you could see what the drop is from here to here and what the increase would be from here to here. And that's shown here. So we looked at, again, our different electrotransfer conditions, our no treatment, and you could see that consistently with the tie-off, we're about 85 to 87% drop in perfusion. And then you can see that no treatment, there's very little difference. If we just inject the DNA, there's also very little difference. And there's two conditions, that's this 20 volts, 20 milliseconds, and 60 volts, 20 milliseconds, where we see a really nice improvement in that area into that ischemic area, with the best being this 20 milliseconds, 60 volts. And you can see that it was about a 68% increase in perfusion from the initial point of tie-off to 2 weeks later. So from this point, we wanted to see, so this was again short-term, and we wanted to see what would happen now if we let these animals go longer. And so the idea was now again to do the procedure the same way, only now because we wanted to stand over with the open chest for more hours. We added another part of it, and that's to do arteriograms before and after the tie-off. So basically the procedure now was to, prior to opening the chest, we would catheterize the swine. We would go in and do an arteriogram of the heart. So again, just like the SPY, we would get a nice depiction of the flow going through all the vessels within the heart. Then we would open up the chest, we would take the SPY picture, we would tie off, we would take another SPY picture, then we would go in again and do another catheterization and see what the block was, then treat, then close up, and then wait and do wait at 2 weeks, and then at 7 weeks would be the terminal point instead of 2 weeks, and see what was going on. So this is the pictures of the arteriogram, and so you could, sorry, maybe if I move this. So you could see here, so this is prior to, so these are two healthy hearts. And I'm not sure, it might be too bright to be able to see this. I know Danielle was saying they tried to turn the lights down, but they couldn't. But you could see the vessels, and then this is right after we tie off. And from my angle, I can't see it from my angle, but I think right here is the tie, or right here. And then you'll see no blood flow coming through there. And then at 7 weeks, we would look at what's going on. And then you could see here in the blow up. So here's the tie off right here, and you see no flow going past that tie. Here where we treated, you could see here's the tie off, and then you could see now there is flow past the tie, and actually here, and if you could see this, you would see that this actually is elongation or new vessel growth from coming off here. So collaterals to here and over here. And I'm not sure if you could see it here or not. But anyway, this kind of, again, gave us a proof of principle that we can actually do this with VEGF-A delivery into the heart and see reperfusion back into that heart. So this is sort of a quantitation of that. So we did 10 animals here and of our electrode transfer, and about 70% or 7 of the 10 had new vessel growth based on that at 2 weeks. And at 7 weeks, it rose up to 8 out of 10 that had it. And this was much higher than either our injection only, which was down at 25 to 30%, or our sham, which was about 40% to about 40% over the 7-week period. So again, it's showing that the concept was valid, and that we could see regrowth of the vessels in there. The other important aspect of this is what would be the last aspect of it, in that can this have an impact on the size of the infarct? So this is the important part. How much damage to the muscle was there? And so after doing this, at 7 weeks, we removed the heart, sectioned and stained, and looked for viable sections and dead sections. And you could see, oops. Here, this is the electrode transfer treated heart, injection only, and the sham. And you could see that here, the infarct area is much smaller than in our controls. So not only was there perfusion that was good to see that we were able to do that, but it actually had a positive effect on the outcome. So this again was very encouraging to be able to know that we could do this and actually have an effect on it. So now the question would be is, okay, so we're there, but we're not actually sure whether VEGF-A is really the right molecule to be doing this. As many of you might know, it's been used in clinical trials and not really seen a great response with it. The question would be is how can we do this? And so we wanted to move to a small model, a rat model, because trying to test different molecules and doing a big matrix in a porcine model is just not feasible to do. And so we shifted to a rat model. We also wanted to look at whether or not we could figure out any better electrode design. So the one that we use to start with now that I just showed you results are obviously worked really well. And it shows the proof of principle that we could actually deliver the plasmid where we wanted it to be, actually get the effect that we were looking for. But the question would be is, can we move this to more of a minimally invasive approach? So if somebody was going in for bypass for a cabbage, they would be, the heart would be stopped anyway, and the chest would be open. It would not be a problem. We could probably use any of these electrodes. But if you didn't want to go through a surgical procedure and you wanted to be more minimally invasive, the idea would be to utilize a catheter. And we've developed catheters utilizing the needle array, but of course you have to miniaturize it. And as with most catheters, they can come out, be unsheathed, and expand out. But with needle electrodes, it becomes very difficult the folding out to make sure that they're perfectly straight and even when you insert them so that the field is the way you want it to do it. So I'm going to show you some new designs that we're looking at that might make it easier to do that. And then also by looking at other molecules, we want to also look and see if this can be used for repair as well as prevention of the infarct. And a lot of this now has been done collaboration with a new young faculty member at the Center for Bioelectrics at ODU. And actually a new electrode design is hers. She has a poster this afternoon, which I would encourage you to go. It's number 156. And I'm going to show you some of this now. So as far as the rat model, so Anya developed this. This is in Sprague Dawley's. And again, still using the 4 needle, but of course now, because it's a rat heart, not a pig heart, we had to sort of make it much smaller to be able to fit in that heart. It's also much more interesting that rat heart rate is probably twice what it is in a pig, which again makes the refractory period much smaller and much more difficult to be able to do it without fibrillating. But rats are also much hardier than pigs as well. So this didn't present a major issue and was able to be resolved. So with this setup, using luciferase and being able to image, and that's also an advantage of the rat, we can get a timeline and image with an IVUS imager as opposed to having to go back in and take the samples. In a pig, we couldn't image the pig in the IVUS. And this can give us a nice kinetic view of what the expression looks like. So again, it worked pretty close to the way it was working in the pig. So we looked at this as a decent model to be able to use to test various different aspects before going back to the pig to see how well it worked therapeutically. So the next thing to look at that we wanted to see in looking at modeling these needles is figuring out what the field is. And for anybody that's looked at electric fields know that the highest field is going to be within these needles, and the lowest is where as the furthest point away from the electrodes. So as you can see, it's not really an even distribution within it. And we knew this going in at the very beginning, and we tried plate electrodes and flat electrodes to be able to, and pin electrodes that were non-penetrating to try and get away from this. But the issue is that the expression was very limited to epicardium, and we really wanted the expression to be the myocardium. And so depth of penetration was an issue. So to get by that, we started looking at this monopolar electrode geometry. And essentially what you have is an active electrode and then a much larger grounding plate, which is at a distant site. And basically what that does is it allows you essentially to have a single electrode at the tissue, at the point of delivery. And your grounding electrode could be someplace else. So even outside the body, for example, in the heart, could just be, the animal could be laying on it. And so this kind of works really well, and when we look at distribution, you could see that we get a much more even distribution pattern. And this is an example of one of the monopolar electrodes. I believe this is the 10 millimeter one. We have different sizes of them. And then the other issue is, what about penetration? And as you can see here, we actually can get pretty good depth of penetration, a few millimeters deep, which allows us to be able to know that we can get into the myocardium and again, still have the field that we want to have as we get in there. So finally, would this work and be able to give us expression within the heart? So again, this is in the rat model. And you could see again, looking at IVUS imaging, that expression of luciferase is there. And interestingly enough, which not necessarily totally surprising, but if we compare it to the results that we got with the 4-needle electrode, compared to the monopolar, it's actually slightly higher. Not significantly higher, but it is higher. And so it's not completely surprising considering the distribution of the fields. So more information on this and also delivery with another molecule, VEGF-B, will be at that poster this afternoon. So in summary, I think I've showed you and pretty much gave you the evidence that electrotransfer can be used to deliver plasma DNA to the heart, and we think this can be done in a controlled manner. We think that, I didn't show you the data on this, but we know by changing the electrode, I mentioned it briefly, that we could we could deliver to the epicardium or the myocardium, and for that matter, even the endocardium, depending on how long our needles are and where we wanted to do it. We can also play with the electrotransfer parameters to give us different expression profiles, either amplitude or kinetics. The one caveat, of course, when you're dealing with the heart, that the pulses have to be synchronized, and they need to be administered during that refractory period in order to avoid fibrillation of the heart. And this, as I mentioned earlier, does affect how long the pulses can be, pulse width of that. And this, when using needle electrodes, doesn't necessarily have that big of an issue, but when we were trying to do non-penetrating electrodes, the longer pulses actually work a little bit better. And that could have been an issue why we couldn't get the penetration with the non-penetrating electrodes. So are also the results that I showed that we could use this to increase the perfusion in the ischemic area of the heart, and this happens fairly quickly within 14 days. We also know that within this that we could also impact the size of the infarct, and this is out 7 weeks after treatment. There is a reduction in that size of the infarct. There's both a porcine and a rat model that allow us to evaluate this. The rat model can be used to evaluate a large number of parameters more quickly than using a porcine model, and then the porcine model can be looked at the translatability of the approach. I showed you two different electrode geometries, although we tested many others, and these were this 4-needle and the monopolar. And finally, I want to acknowledge all the people involved. Dr. Bulashiva, who has a poster this afternoon, I mentioned her earlier. Dr. Hargrave did a lot of the surgeries along with Dr. Strange from the Naval Hospital in Portsmouth when we were doing the porcine model. Dr. Murray was the veterinarian at ODU that helped us quite a bit. And then other people that were involved, Catherine and Annalise, in evaluating samples, and then our vivarium staff. And I also want to mention Dr. Marshall back many years ago when we were both at USF. We kind of talked about doing this kind of thing and started it, and he was instrumental even when we were starting at ODU being able to do this. And then, of course, the funding, we couldn't do this without our funding on that. And then one last thing, just a quick plug. This July, we will have a fundamental and applied bioelectrics workshop. If this is of any interest to you, this is a lecture and hands-on wet lab on how to do various different things with electric fields in biological systems. And I thank you all for your attention.
Thank you very much for this very innovative technology that could open really new doors to the field. Maybe, yes, please. Can you go to the microphone?
Have you been able to identify which cells are the most permissive to the treatment, cardiomyocyte or non-myocyte cell?
Yeah, that's a good question. So we do know that we do get delivery to cardiomyocytes. We have seen that, and actually there is a picture on the poster that would show that. But I will also say with the caveat that this is not specific delivery to specific cell types. So we haven't yet found conditions that would actually target specific cells. So it's more targeting a particular area and the cells within that area. But definitely cardiomyocytes are expressing the plasmid.
Yeah. How about fibroblasts?
Yes, I believe that, yes. And I'm trying to think if there's a picture of that on the poster or not. There might be, but yes, they are there. You could see them, and there is expression within there.
And I'm just curious about the expression drops sort of gradually over 2 weeks, which, I was just wondering, is that just a general property of this technique? Or do you think what's really going on there? Is this silencing of the transgene, maybe the bacterial sequences in the plasmid, or is this just going to happen with kind of any DNA you deliver?
So, good question, and we haven't really looked at that, but I believe that it is probably due to the plasmid. It's probably a silencing of the plasmid. We haven't investigated it. We were just happy that it was short-term. We didn't necessarily want to change it to make it longer-term. But it's a good question, and I would only be guessing on specifics as to why that is. I don't think it's specific to the technique. It may be specific to that particular tissue or the plasmid, because we could use this approach to other tissue and we can get expression out for months. So it just depends on various different things.
Congratulations on your work in a challenging animal model, the swine model. I just wanted to ask about the figures that you showed with respect to the VEGF expression, and it was actually showing quantity in picograms. How are you measuring that? In a specific sample size, or?
So yes, good question. So that's actually total. So what we would do is we would take a punch biopsy of the area that we treated, and then homogenize that whole sample, and then based on the weight of that sample, and then we would do it. So we decided it became a better measure to just put it as total as opposed to per weight of the sample, since they all were pretty close to the same size in that.
Okay, very good. Have you measured the fibrosis decrease or not? Because suppose this increase the angiogenesis, so this would be decrease the fibrosis, right?
So you're asking me about fibrosis within the treated area or
Yes.
in the infarct area?
Yes, you reduce the fibrosis in the infected area or not?
Yes. So I showed the one slide, maybe I went through quickly, but yes. So it's not eliminated, there's some there, but it's reduced.
Two short questions and 4 short answers. With the plate electrode, with the plate monopolar electrode, you still need to administer the DNA into the tissue. Yes. So you still need needles, right? So what would be the advantage of having plate electrode in addition to the fact that you still have to distribute evenly with many needles DNA into the tissue?
So yeah, another good question. So the advantage would be it's one needle versus 5. Second, the field distribution is much better with the plate as opposed to the needles.
Yeah, but the DNA distribution with one needle would not be evenly distributed.
Well, yeah, and there are ways to do the injection in a way that you can distribute it a little bit better. But you're right. I mean, it's going to be a combination of the two things: how your injection is distributing the DNA and how the field is distributed. The other aspect, which I mentioned very quickly, was that when we moved to the catheter device, it's much easier to do the plate than the needles when it folds out. So
Okay.
that's another aspect of that.
And don't you think, because in the pig, in the swine, what was the distance between the electrode? Which was the distance?
5 millimeters.
5
And the
millimeters.
needles were 5 millimeters.
5 millimeters. Yeah.
5 millimeters square.
Yeah. So do you think it could be possible if you decrease the distance between the needle to lower the field? It's a classical electro transfer law. That
Well,
if
you can lower the applied voltage, but the field is the same. And we did that.
Exactly.
We have a 2.5 millimeter array as well. The problem is what it turns out, I mean, I kind of joked about it with doing the 4 sites, but for whatever reason, that's what worked. Not just the 4 sites, but the volume within each of those sites. So the 5 by 5, the 25 millimeter square area in those 4 sites turns out to be really work well. When you cut that, reduce it to a 2.5 by 2.5, you now have to treat more areas. So that's more pulses, more chances for fibrillation, and so
I see.
that's why the 5 by 5 seems to work.
Okay, thank you very much. Thank you again.